differentiation, proliferation, maturation, so that we are able now to have a fully specialized cells components. So interleukins are usually signaling molecules of immune cells, which usually are part of our body cells. So interleukins are just enzymes, they're proteins. They help in transduction and sending essential communications to each and every progenitor cell so that they're able now to be specialized to divide and differentiate into specialized cells. The interleukins also are able to work in conjunction with the growth factors to initiate further proliferation, differentiation, and specific cell lines. And sources have mentioned that there are over 35 well-known interleukins. So I want you to go and check around 9 of them can be part of section B in your exams. Go and check what is the function of IL-1 in relation to hematopoiesis, IL-2, IL-3, IL-4, IL-5, IL-6, IL-7, IL-8, IL-9, and IL-10. Very critical. Go and check in that and their relationship with individualized growth factors towards influencing different cell lines. Are we clear there? Tuko salama? Are you getting me? Yes? Are we together?
Yes.
Yes.
Okay. Yes. So I want you to go and check on that so that now the next class you are able now to be at par and you familiarize yourself with them. So go and check on the functions of the interleukins and growth factors on how they influence the differentiation, proliferations, and maturations of these stem cells. Because we have over 35, so look at around 10 of them. Okay, another important component that usually influence the hematopoietic nature of the stem cells is the growth factors. And the growth factors is usually encoded by a single gene. I told you that growth factors are just examples of codal examples of proteins that are usually translated. You guys have done dogma theory, yeah? That is how the genes are usually transcribed into mRNA within the transcripts, what we call the transcriptomics. And now from there, they're being translated into an individual protein. Individual protein here now can be a collection of hub proteins called growth factors. And an examples of these genes that usually encode for growth factors, an individual is erythropoietin, which is located on chromosome 7. And these genes usually are found on cluster on long terms of chromosome 5 or chromosome 5 or 7. The major roles of this is that they usually regulate proliferations and differentiations of hematopoietic stem cells. They also influence the survival and functions of mature blood cells. Now, just to pose a question, this one needs critical thinking on how. How do you think the growth factors can influence the survival of independent hematopoietic stem cells? Yes? How can it influence the survival of hematopoietic stem cells? Yes? Any take? How can yes, Polycarp? Yes,
They regulate proliferation and differentiation of the hematopoietic cells.
Okay, that's a trial. That's a nice one. But overall, how do you think that they can influence the survival of these stem cells? Because for sure, the nature of hematopoietic multipluripotent stem cells, their nature, it has been known that they usually have self-renewal capacity. That means the original pluripotent cell has to divide again to the secondary one. So that means they have to retain the original version. So how does it influence the survival overall? Yes. How? How does it influence? Yes, Abigail. I can see Abigail. Abigail can tell us how these growth factors usually influence the survival of HPCs. Some are typing, they preventing apoptosis. Uh-huh. Okay, that's a nice take. That's a very nice take. I don't know. Have you guys done immunology or physiology? Yeah. Have you guys done immunology
Not yet.
or physiology?
Yeah, we have done.
Okay, then if you guys have done the fundamentals of physiology, then you guys ought to tie each and every relationship between the growth factors and the individual progenitor cells, how they influence differentiation, how they influence survival, how do they influence maturation. It's a very critical aspect that you ought not to bypass as you're reading. Okay, so they regulate survival and the functions of the mature blood cells. And these growth factors are usually being tested and they're usually used as very significant profiles in clinical trials and in treatments and in aspects of hematological disorders. For example, we have the EPO, what we call now the erythropoietin hormone, what we call now the granulocyte monocyte cell line differentiation units, yeah? The CSF. Then we have the granulocyte CSFs and the monocyte, yeah? And the IL-3. I wanted to pose a question, but now you guys have not done immunology, and definitely now that might be a question for the next lesson. Yeah? So they are also been used in adjunct therapy. So a certain question might be posed. What do you think might be the applications of these growth factors towards the neoadjuvant in the clinical field? Yes, what's your take? In the clinical neoadjuvant field, what do you think might be the one of the applications of growth factors? Yes. Any take? Kiprop? Kiprop? Any take? You can just unmute and talk. You can unmute and talk. Yes? Abby, Selena. Selena. Okay, I can see Boniface is saying that he doesn't have any idea. You guys are supposed to be checking on that. You must read very widely, yeah, so as to you tie the application aspect of these growth factors to the neoadjuvant in the clinical aspects. Yeah? So we have now the the mature lines of individualized blood components, such as the T lymphocytes, the monocytes, and the macrophages, and even the stromal cells, which are usually called the NAS cells, are usually aiding in ensuring that we have overall survival and maturation of these cells as they divide along their cell lines. And you find that now the sources of growth factors are there with an exception of 90%, which usually synthesized in the kidneys, and thrombopoietin, which is usually made large in the liver. Because I know you guys have done anatomy, you're able to tie on how these different EPOs and different cells are usually being manufactured as per the organ systems in the anatomy. Are we okay? Those are the features of the hematopoietic growth factors. You must know all of them. Number one is that they usually regulate growth and differentiation. That is their optimal feature. There are a family of glycoproteins with clinical utility. And I want to emphasize on feature number two, that they are a family of glycoproteins. Why do you think? That being that they belong to a family of the GPs, yeah, the glycoproteins, they're now influential in clinical diagnosis. Yes? Any take? Why do you think now they are very influential in clinical diagnosis? Yes? Yes? Any take? Wamukoya, there's somebody called Wamukoya, Chris. A trial, a family of the GPs with clinical utility. So why do you think it's very critical? Yes? Crispin Baraza? You guys are medics, you're supposed to be having these at your fingertips. Application questions must always incur in every section. So you must always get ready. You have said that one of the most critical feature of these growth factors is that they are family of the glycoproteins. So why do you think? Tie that to its clinical utility. Yeah? Yes, nurse. There's somebody carrying up his aunt. Yes, the nurse. Yeah.
Okay, the clinical utility means that they are used in medical treatment or diagnostics.
Okay.
So they are often used in medicine to stimulate the production of blood cells, especially in patients who are undergoing therapy, radiation therapy, or those with certain blood disorders. I think.
That's nice. That's a nice take. That's a nice take. Okay, but you've kind of generalized it a bit, definitely. The answer lies between the glycoproteins. Yeah, answer lies in the glycoproteins. They're not a family of lipoproteins, but they're a family of glycoproteins. So it's very essential for me to stress that so that you guys are able to get that. So you guys got it. What are the functions of every glycoproteins on the nature? You have done the anatomy and the composition of the cell membrane. You have done the structure of the virus, the HIV. They have GP120, GP21, GP41. What is the essence of that in clinical diagnostic utility? So you go and check on that. There's a nice trial from, that's your name, Mass Club. The nice trial. Yeah. Okay, they also have different specificity. Yeah, they have specificity. Okay, what do you mean by specificity? Yeah. What do you mean by specificity? Yes? What do you mean by specificity? Faustin. Faustin. Faith. Faith Howie. Faith Howie. What do you mean by that? Faith Nabwire. Okay. Okay. You guys go and check on that, yeah. I will not tell you that, yeah. They have what is the difference? Because you must know when they say that a test is highly specific or a test is highly sensitive, sensitivity and specificity, one and the different. Okay, they mean different things, but definitely you have to, because for you now to get the sensitivity, you have also to understand what we call specificity. Yeah? You go and check on that. The next feature is that they contain different origins and they bind to cognate receptors on progenitor cells. And the last feature is that they act locally or they act at a distance. And that is where now it's very influential on several cytokines for signal introduction and sending of communications to the distant bone marrow to effect for the production of the blood cell components. Okay. The next one is that they regulate proliferation and differentiation, and they usually prevent apoptosis. What another gentleman had posted, that is one of the critical aspects on how these cells usually survive. Yeah. Then they enhance functions of some end-stage cells. Okay, maybe just to pose another question to the entire class. I need this class to be very interactive. That is how now you can know. You don't learn science by keeping quiet. So how can these growth factors prevent apoptosis? Because them preventing apoptosis, that means they'll enhance the overall survival of individual cell components upon maturation. What's your take on them preventing apoptosis? In one word, can someone just explain in one word? How does it prevent apoptosis? Yes, George William, yeah? And see, very, very nice name called George William. Yes? Yes, Nas Club, I can see your hand raised.
Okay, the growth factor can prevent apoptosis by activating the intracellular surviving signaling pathways in hematopoietic stem cells.
Yeah. And that's a nice one. That's a really nice one. They activate or they dysregulate. Yeah, they definitely usually do what we call deactivation of negative regulators, yeah, or negative loops towards now preventing apoptosis and enhancing survival, overall survival of these individualized cellular components. That's a nice one. Any other take? Glorious? Yes, Kisa is carrying up. Yeah.
By survival, they act as survival factors inhibiting cell death.
Okay, how? That's a nice one, but now you have to tie it to a very strong scientific statement. Yeah, just as the way NAS, I don't know, is that your name? NAS's club. That is how definitely, yeah, how they dysregulate intracellular pathways and overall now being cumulative of the negative feedback loops. Yeah. Were, yeah, but his name is Were, yeah, and that's a nice take from Were. Yeah, Hisa. Okay, that's a nice one, but definitely have to tie it with another scientific statement. Is a good trial. Any other trial? How can they prevent apoptosis, thus now influencing the overall cumulative survival of these cells? Of course, I can't leave out the class rep, yeah? It's called Levy. Levy must talk because Levy ni kiongozi. So definitely she must lead by an example. What's your take? Anaitwa Levy ni kiongozi, yeah? Patrick is saying that after binding to growth factors, they enhance growth of cells. Yeah? That's a nice one. But you have to tie it to apoptosis. Yeah? They block pro-apoptotic signals. That's a nice one. That's a very nice one. You know? Yes. Any other person? I don't want types. I want you to talk. I want you to talk. Yeah. They enhance functions. You've heard what you call the TP53 in physiology. You've heard of what you call the BCL1, BCL2 in physiology. You've heard what we call the cytochromes, the ubiquitinone pathways. You've heard of that? Hey, guys, have you done physiology? Yeah? Where? Confirm with
Yes.
me. Have you guys, you guys have done physiology. I'm precious. You've done physiology. And you guys must
Yeah.
definitely, you guys go and check the relationship between that. Yeah? How they can usually prevent apoptosis and thus influencing now the overall survival of these cells. It's very critical to check on it. Nice one. Okay. On the aspect parts, we have the examples of the cytokines, which are usually IL-1, IL-3 up to IL-9. But since that you guys have done physiology, you know the functions of IL-1, you must know the functions of IL-3, you must know the functions of IL-4 in relation to the hematopoietic stem cells maturations to individual components. That is the RBCs, the white blood cells, and the platelets. Yeah. The tumor growth factors beta. Yeah. So. Are we clear there?
Those things you are mentioning, maybe you are hearing them for the first time.
Yeah. You're hearing them for the first time.
Yeah.
That's Newton. Newton, you guys have done physiology, so you're able now to try. You must try because most aspects of these functions of the factors that usually influence hematopoietic growth or stem cells growth, the words now being individualized, blood components, they're usually majorly tied with the physiology. You can explain them because now if you're supposed to get it in section, say, you explain how apoptosis influence the maturation and differentiation and survival of hematopoietic stem cells. That is not taught everywhere. That is not taught anywhere. Yeah. So you ought to have checked that. Yeah. Yeah. If I say that you explain on how IL-1, IL-3, and IL-10 influence the process of hematopoiesis. Yeah. So it's not taught anywhere. Just personal initiative on how you dig into your websites and how you check it. Yeah. So that's very influential. Yeah. That's very influential. Nice. So the next thing is that I'll definitely project what you call the overall hematopoietic stem cells, the overall process of hematopoiesis, and is usually starting with what we call the CD438. So CD34 positive cluster of differentiation. That is now what we call the overall primary progenitor cells. In other terms, it will be called hematopoietic multipluripotent hematopoietic stem cells, abbreviated as HMPSC. Main feature of progenitor CD34 is that it must undergo self-renewal in the first place. That means it divides itself so that now the original progenitor, the CD4, the CD34 positive, remains in the bone marrow. The secondary CD34 positives now undergoes what we call now they divide into progenitor cell lines, and which is usually the cell formulating units or the cell colony formulating units, that is under erythroid nature. Then we have granulocyte monocyte cell formulating units, which will now dictate on the differentiation and overall survival of these granulocytes and monocytes. And then we have the monocytes and the K cells and the platelets self-formulating units. Now what we call the myeloblasts and the lymphoblasts. Those are the main progenitor cells. So the myeloblasts will definitely divide into three colony forming units. You're able to see as projected. And the lymphoblasts usually undergo straight proliferations and direction to precursor cells. So I want you to go and study that process very keenly, and definitely now this diagram is self-explanatory. How the precursors of erythroblasts actually from myeloblasts they go to erythroblasts. Yeah? The colony forming units erythroblasts will undergo what we call the pro-erythroblasts. Go and check on the individualized characteristics, and when you're checking on the features of these precursor cells, take note on the nucleus, take note on the size, and take note on the chromatin individuals. I hope you have a pen and a paper where you are writing. Take note on the nucleus, the size and the content. Take note on the secondary granules and the primary granules initiated. Take note on the nuclear size and the cytoplasm. And what you call now the contour irregularities. Then it goes to basophilic erythroblasts. Then it goes to polychromatophilic now. Polychromatophilic now being that now it has a mixture of nucleic acid, the DNA and the RNA. So definitely takes different colors of erythroblasts and how it matures straight to erythroblasts. That is in the under PBF, the peripheral blood fields. So I want you to go and check on that very keenly. If you're able to master that, then you have your free 20 marks in section C come your end of semester exams. So that one to mean hiyo lazima italetwa. Yeah, yeah, hiyo lazima italetwa. Literally. Yeah, so go and check on that. If you're able to, if you are good crammer, cram it, but you are also able to understand. You must try to understand because it will form part of you for the rest of your medical life. Yeah? A very critical aspect. You understand the normal physiology. You have to understand the normal hematopoietic stem cells process. You have to understand the individualized features of these precursor cells. So as now, when you guys will be learning on how pathology and everything, you're able now to tie the pathological aspect of these individual cells to the normal physiology of them. And that will now better your understanding. Yeah? Very critical. Very critical. Nice. So I'll send you the notes at the end of this reflection. That is just the diagrammatic explanation. You're able now to check on that, individualize the morphological aspects on how they behave. You're able to see. So I'm going to share with you guys on this one, and maybe just to strengthen on this validation that I'm going also to look into the notes on how I'm able to synchronize the individualized precursor cells and the mature cells in the PDF and how they're being viewed morphologically, so that you are able to have a deep understanding on it. But the most critical aspect here in this lecture is that, number one, get to know the meaning of hematopoiesis as a summary. Number 2, get to know the different types of hematopoietic growth factors visually influencing this hematopoietic process. You have to understand what are these examples of the growth factors, what are these examples of the interleukins, what are these TGF, what we call the tumor growth factors beta. And you have to be able to understand that. And overall, the most important is how does now the progenitor cells, the HMPSC, now undergoes differentiation up to the survival age, up to the maturation stage, where now we have the maturations of now erythrocytes, maturations of now the lymphocytes, and maturations of the platelets, or what we call the thrombocytes. Sawa sawa. Are we clear? Okay, so that is what I literally wanted to share with you today because that's a small lecture. I usually prefer, my voice is breaking, but we are almost ending the lecture anyway. So I usually prefer physical sessions, so I will strive and organize with you guys course of next week so that we are able now to crush a lot of information because is it usually on Monday or on Tuesday? Yes,
On
is our
Tuesday.
lecture on Monday?
On
On Tuesday. Okay, on Tuesday. Okay, I'll literally check next week if on Tuesday is from 7 or 7:30.
7:30.
7:30. It goes 7:30 to 3 hours. Yeah.
Yeah.
Okay, so that's mean from 7:30 to around 10:30 for 3 hours. So literally, we can check on that. Where it's telling me we can organize for online apart from Tuesday. Okay, we can always fix if we can able to crash Tuesday or Wednesday because Levia told me that you guys are always quite free on Wednesday around 2 p.m. Yeah, so literally always compensating on around that time. But I also check on how now we can organize also on a physical session on Tuesday morning hours, yeah. So that we're able now to learn a lot. But by 15th of June or late 20th of June, we are over, we are done with this. And then I can leave you now to read and do your cuts. Yes, I can see Chris Omukoya carrying his on. Yes.
Okay, I have a concern about the admission of the into this forum.
Yeah.
I think you are supposed to give us at least like 10 minutes before you start for people to join in. So like a person like me, I join in late. Since I clicked in the link at exactly at 1:55 and you are not around. So I gave up. So after I guess I joined in at exactly at 2:10 and you're already at in between the notes. So I was confused.
Oh, okay. That one is taken care of. The next time we'll be having this session is that I'll be giving you approximately 5 minutes to join. Yeah. So when the session is supposed to be starting at 2 p.m., then definitely now you guys around 2:05. Yeah. So that you're able to join. Yeah. That's taken, Chris. That's noted. Any other query? Any other feedback? Any other question? Yes. No questions from 67 medics, 67 plus medics. No question, no concern apart from Omukoya.
Okay, I have another concern
Yes, Omukoya.
about the yes about the previous you are given the course outline,
Yeah.
but as per the course outline, this I guess this is the first lesson that you're having.
Uh-huh.
So I have not seen the first subtopics, that is the introduction to hematology and the blood constituents and their functions.
No. Don't worry. You just I gave you the first. Did Levi send you the first notes, the first lecture?
Yes.
That's awesome. The second lecture is this one, hematopoiesis. So this the next lecture now we are going to check into now the individualized blood components. We have to move chronologically first. So also, so don't worry
Okay.
about it earlier. We'll tackle everything that is usually within that course outline. So you guys don't worry.
Okay.
And nice one, Mukoya. Any other intake? Yes, I can see Victor.
Okay, thanks for this opportunity. So,
Yeah.
sir, there's something you talked about. I just heard that check on nucleus, secondary and primary granules, size, chromosomes, and nucleus. So, I would like you to clarify on that.
Okay, Victor, thank you. Okay, what I've said is that when you're checking on the hematopoietic stem cell process or the process of hematopoiesis, take note on the morphological features and the examples of the morphological features. How do they change from the progenitor to the precursor cells to now where they mature? Check on the nucleus, check on the cytoplasm, because that is where now majority of the this morphological feature usually changes as they transcend to the transition between now the progenitor cells to the mature cells. So take note on the nucleus, take note on the size of the nucleus and the components of the nucleus. Take note on the cytoplasm of these cells. Take notes of the central pala in terms of now the RBCs. Take notes of the chromatin nature. Take notes of the nucleic acids. And finally, take notes on what we call the granules, the secondary granules and the primary granules. So yeah, that one will aid you in diagnosis actually. It'll aid you in diagnosis, so it will form part of you for the rest of your medical life in your 4 years. Yeah. Any other intake? Yes, Baraza.
So my
Yes.
in the process of hematopoiesis is my doing the same thing in any form of
You're breaking, Christoph. You're breaking, Baraza. I don't know if the rest of the class is getting you, whatever you're saying, but you're breaking. I don't know. Can you come up again? Yeah.
actually in the process of hematopoiesis is myelopoiesis means the same thing as the clinical hepatocyte.
Is myelopoiesis mean?
Means the same thing as the colony blastocyte.
I don't know. You're breaking literally. I don't know. Can you type so that maybe I don't know? Can you get? Because people are not getting you literally. You're
Okay.
breaking. Yeah, you're breaking literally. Just type and I'll definitely see on how to go about it. Yes. Any other concern? Any other query? Any other question? Kiongozi hajaongea. I know only of Levy. Who is the male rep? Male rep anaitwa nani?
He's called Chris. He has already spoken.
Okay, that's awesome. That's awesome. Okay, okay. So you guys, you send me the. I'll definitely send you the notes. You go and check into it. Any query, you definitely can reach me out, so that now in the next session you're able to address it. But for the next class, we are going now to check a very simple topic on the functions and different components of this blood component, yeah, and to deep down into the RBCs, the lymphocytes, and the platelets, and check on their physiological importance and also their clinical importance overall. Sawa sawa. Are we okay? Okay, if there is no, thank you for that, Boniface. Crispin is asking, in the hematopoiesis process, is myelopoiesis means same as colony forming blastocyst? Definitely that is true. Yeah, definitely that is true. You have your AI, your website, so definitely you can check that. That's definitely very true, Baraza. Yeah. Okay, so if there's no any concern, then allow me to end the meeting here. And you guys have a blessed day, a wonderful day ahead. Thank you.